You're watching a tooltip video for the US EPA's CompTox Chemicals dashboard. These are short tutorials for individual tool features. More tooltips and longer training materials are also available to you as part of the New Approach Methods or NAMS training website. This video provides a brief overview of searching by assay or gene within the dashboard. We'll start at the homepage of the dashboard and select the Assay Gene tab above the search bar. This is a good place to start if you're interested in exploring chemicals that target specific genes, for example. Here, we can type in the name of an assay endpoint or a gene of interest. Let's say I'm interested in the transcription factor EGR1. If I type this in the search bar, the dropdown auto-populates with matching terms. In this case, I can either select a specific assay endpoint or click on the gene name. Let's click on the first assay endpoint. Here at the top of the page, we can read a description about the assay and also see the number of chemicals that have been tested, 1,057. Below this is the detailed assay information bar with all the relevant technical details on the assay. Down here in the results field, we are shown all chemicals that were active in the assay. In this case, there were 97 out of the total tested. If you wanted to know more about this specific group of chemicals, we have the option of sending these to batch search. We can also apply filters, such as removing the multi-component chemicals before conducting the batch search. As in other areas of the dashboard, we can change our preferred view by selecting either the tile or the grid view. This preference will be saved for the next time you access the dashboard. Another feature of the results page is the histograms tab. By expanding this tab, we can see two histograms. The one on the left bins the active chemicals by log AC50 value, while the histogram on the right bins the active chemicals by scaled top value. Hovering over the bars will give the range and the number of chemicals within each bin. Let's now return to the homepage search bar and select the gene rather than the assay endpoint name when we type in EGR1. This page shows all individual assay endpoints that target our candidate gene. You can see there are 4 out of a total of 2,205 assay endpoints. We have the option of filtering this list by vendor by clicking the Filter drop-down menu at the top of the screen. We can also further refine the list by using the filter option in any of these columns. Clicking this paper icon next to the endpoint name will bring you to the detailed assay information page and gives you the option to copy or export. You'll see that the third one on the list is the endpoint name we had just explored. Selecting this hyperlink will bring you back to that page. This ends our brief exploration of the dashboard's assay or gene search function. Thank you for accessing tooltips for the CompTox Chemicals dashboard. You can find more tooltips for this tool and other tools on the NAMS training website.